Right. So, does anybody have any questions from the last week?
No.
No one? Okay. So in the last session, we actually started to look at the lymphatic system, and we started to look at the organs involved in the lymphatic system, like the spleen, the lymph nodes, the thymus, and the lymphatic nodules. And we also looked at the first line of defense, which is your skin and mucous membranes, right? So we will continue to explore that further. You already have access to the screencast, so I will go into details of the major concepts. I won't go through each and every slide, but the main things that you need to know about the concepts. And if you have any questions, feel free to ask me and I can answer them, right? So today's session is about the second line of defense. So when we talk about the second line of defense, now these are all the chemical and cellular responses. These are more like the processes of inflammation, phagocytosis, and the different antimicrobial substances that our body releases to work as chemical signals, you know, like to signal our body about the intruders or of the infection or something is wrong with the body, basically. So today's session, the main points, main key aspects or the focus areas are basically two major processes, inflammation and phagocytosis. Why are they so important? Because whatever happens in your body, whether it is a, say, injury, whether it is infection, whether it's a response to allergies or to autoimmune diseases, whatever, inflammation is the basis of the immune response. Okay, it will always trigger inflammation in some way or the other. So it's very important to understand what is inflammation, why it happens, how it happens, and basically what are the different molecules that participate in creating that inflammatory response. Okay, and whenever there is inflammation, particularly if you are dealing with infection or injury, you will also have some degree of phagocytosis, which is basically removing the dead cells or removing the bacteria or the pathogens that have invaded the body, or in other words, removing the cells that are not normal or that are infected or that are injured or that are dead. Okay, so those two processes are the ones that we will primarily focus on. Of course, I will also talk a little bit about the other methods of second line of defense, like the complement system, the antimicrobial substances, and things like that. Okay, so let us start with the second line of defense. So second line of defense, it basically includes the antimicrobial substances, the natural killer cells, the phagocytes, the inflammation, and fever. These are few processes that are involved in this line of defense. Now this is, again, like the first line of defense, this is an innate immune response, which means your body already knows how to trigger inflammation or how to trigger phagocytosis or how these antimicrobial proteins should be working. You know, it doesn't have to learn these things. It is inborn. It is something that you're born with. Okay, so let's start with the first one, talking about antimicrobial substances. So the word tells you what it does, right? Anti as in oppose, microbes. So these substances, they help in some way, they help to fight the pathogens or the microorganisms that would have otherwise invaded your body system. Okay, so what is included in here? So antimicrobial substances, it basically includes your interferons, complement system, iron binding proteins, and antimicrobial proteins produced by different body cells. Okay, so let's look at each of these in detail. Now talking about interferons, the word tells you interferons. So these are protein molecules which are produced by the lymphocytes or the macrophages in the tissue or by the fibroblasts and other neighboring cells of the tissue. So fibroblasts, if they are infected with the bacteria or parasites or something like that, like a pathogen, they will be producing these interferons, right? Broadly speaking, there are three categories of interferons. We have alpha interferons, we have beta interferons, and gamma interferons. It obviously depends on the pathogen that has invaded your system, like a virus or a bacteria or a parasite or a protozoa. Depending on that, different interferons are produced. Okay, so what do they basically do? Now, interferons, they are kind of proteins which will help to make antiviral proteins or antipathogenic proteins, particularly viruses. Most of the viruses will trigger interferon production in the body, and they will release interferons to, the body will release interferons to fight with the viruses. Okay, some bacteria also produce interferons, like say tuberculosis, for example. So tuberculosis, it involves a specific immune response, but interferons participate in that specific response, and they make gamma interferons against the protein produced by that bacteria. So they are a part of both the second as well as the third line, in a way, you can say that. But primarily, second line of defense. Interferons are something that your body already makes as a usual process, and it knows how to use it. So it doesn't have to wait for a specific exposure or a particular type of a like a bacteria or a virus or things like that to trigger the response. It's already there. Okay, so interferons, they are basically meant for interfering or stopping the viral replication most of the time in the common viruses that you come across, like the adenoviruses or the rhinoviruses, enteroviruses, the common infections that we come across, the viral infections, right? So that's about the interferons. Then you have the complement system. So complement proteins, they are actually a group of proteins. They're not one or two proteins. There are about 30 proteins, which are usually, they are inactive. They are present in your plasma. They are present in your blood. They exist as the plasma proteins. Some of these complement proteins are also present on the surfaces of your body cells, like your tissue cells, your macrophages, like that. Okay. What do? They basically activate the immune responses. So they complement the immune responses. So they help in activation of inflammation or activation of phagocytosis, or maybe even diapedesis, which is bringing the lymphocytes or the white blood cells into the injured or infected tissue, right? So migration of the white blood cells. So it helps with that kind of responses. So it doesn't actually participate directly in any immune response, but indirectly helps the immune responses. Either it speeds it up or activates them. So that's why it's called complement proteins. And these complement proteins, they work in a cascadic manner. So one protein will activate few others, they will activate few more, and like that. And that's how basically they work. Okay. Complement system is again very important when it comes to allergies and specific immune responses. Also, a lot of specific immune responses are also activated by complement system. Okay. Then we also have certain iron binding proteins. So iron binding proteins. Now, you know, there are bacteria that kind of love iron. So they thrive on iron-filled environment. There are also certain pathogens that love iron and they thrive on this environment, right? So iron binding proteins, they will prevent that free iron from being available to the pathogens, and therefore they inhibit the growth of bacteria that rely primarily on iron. Okay, so there are a lot of iron binding proteins, and as such also iron is a metal that easily rusts in presence of oxygen. So you don't want too much free iron circulating in your blood anyways, right? So the proteins will either help to transport the iron from one organ to another, like for example, during recycling of red blood cells, we make a lot of iron from breakdown of the red blood cells in the spleen. Now this iron needs to be stored somewhere. So transferrin is one of the iron binding protein which will help to carry this iron to the organs where it will be stored for future use. Okay. Similarly, you have lactoferrin in the milk, you have ferritin, you have hemosiderin. So these are all different iron binding proteins which are present in the blood or in the tissues like your spleen, liver, and bone marrow, which are primarily either metabolizing the red blood cells or producing the red blood cells. So they're either using the iron or they're storing the iron. Okay. So hemoglobin, again, an iron containing pigment, and it's the core part of your red blood cells, as you have already learned in hematology, right? So that's also an iron binding protein. So presence of these iron binding proteins helps us to inhibit the growth of certain group of bacteria which use iron as their source of energy, as their source of metabolism. Right? Then you have other antimicrobial proteins which are produced by different tissue cells. We learned about some tissue cells like the dendritic cells, the mast cells, etc. in our tissues, in the macrophages in our tissues. So a lot of these cells, particularly immunologically active cells like the mast cells and the dendritic cells, they will produce antimicrobial proteins. Okay, so for example, some of these become the part of your bodily secretions, like in the sweat. You have a lot of antimicrobial proteins in the sweat, which is produced by your skin, and thus it prevents the growth of bacteria on the skin. Right. Similarly, you have defenses and cathepsins, which are produced by your neutrophils, macrophages, and the skin cells. Again, helping with the defense mechanism, helping with the inhibition of the bacterial growth on your skin or in your blood or in your tissues. Okay. Thrombocytin is a platelet-based antimicrobial protein that fights the bacteria and prevents the growth of bacteria during the blood clotting mechanisms and things like that. So a lot of different antimicrobial proteins are produced by your body, which work in some way to reduce or restrict the growth of bacteria, okay, or any pathogens, viruses, bacteria, etc. So these are all chemical molecules. So we can say that in a way they are all signaling molecules. They signal our body to either initiate an immune response or to speed up an existing immune response, or even to limit certain responses and therefore limit the growth of bacteria or viruses or replication of these things in your body. Right? Then we move on to the cells that are involved in the second line of defense. Apart from our normal phagocytes like the macrophages and the neutrophils and lymphocytes, right? We also have natural killer cells. Now, natural killer cells, they are a type of lymphocyte which are considered to be non-specific. They are a part of cytotoxic lymphocytes which become the part of your second line of defense, the non-specific line of defense. Okay. Natural killer cells are generally present in the organs that are directly or indirectly involved in the immune response. So that will be your blood, your spleen, your thymus, your bone marrow. Right? So those are the primary organs where we find the natural killer cells. So how are they different from the other cells? Okay, natural killer cells, they will be activated in, they have a non-specific response, but they are activated in a more rigorous, more serious type of an infection. Like say when the neutrophils or the phagocytes, the normal phagocytes are unable to combat with a particular pathogen, then they will need the help of the natural killer cells. So we can put it that way. So they attack the body cells, particularly in cases where you need a second line of defense, you need a innate defense against stronger antigens or against stronger pathogens. So also against some tumor cells, because tumor cells, they are from your body cells. So often they will also trigger a non-specific response to start with. Okay. They work by causing cytolysis, and that's why they are called natural killer cells. So they kill the cells. They release proteins, they release chemical substances which will initiate cytolysis or apoptosis, cell death, right? So programmed cell death. So they induce apoptosis or cytolysis of your body cells. Okay? They are also used by the phagocytes to kill the bacteria or the pathogens that would have entered our body system. Okay? So all these are your initial stages of your second line of defense, right? Then we come to the mechanisms that are involved in the second line of defense. Okay. Now, before I go into the phagocytosis and the actual process of phagocytosis, anybody has any questions around the antimicrobial proteins or the natural killer cells? No questions?
No.
Okay. All right. So let's go into phagocytosis now. So phagocytosis, does anybody remember what is phagocytosis? Have you heard of this term before? You know, when you learned your biology 1, you learned about cellular transport mechanisms. And one of the ways the cells transport things in and out is through the vesicles. And phagocytosis was one of the vesicular transport that the cells use to bring another cell or another solid substance into the cell for digestion. Okay, so in other words, we can say that phagocytosis is cell eating, right? So it's a way of the cell to get rid of the microorganisms or the macrophages which are presenting with the specific antigens, right? So which cells can carry out phagocytosis? Primarily, there are two cells that have a very good capacity to carry out phagocytosis. We have the neutrophils, which are your polymorphs, and you have the macrophages. So neutrophils and macrophages, they are the primary phagocytic cells. They carry out phagocytosis. So when we talk about the macrophages, you know, there are two types of macrophages. There are macrophages which are in the tissue and they remain in that particular tissue. So they are called fixed macrophages. And there are macrophages in our blood. When they are in the blood, we call them monocytes. Okay. Now, many of these monocytes, when there is infection in our body or when there is injury in our body, many of these monocytes will leave your blood vessels, your blood capillaries, and enter the tissue spaces. So these macrophages, they are called wandering macrophages. Why? Because they are circulating in the blood and they will come out into the tissues as and when required through the diapedesis, immigration process, right? So neutrophils and macrophages, these are the two cells that carry out phagocytosis. So let's have a look at the phagocytosis mechanism, okay? So phagocytosis involves 5 steps. So just let me bring that up, okay? So your first step is chemotaxis. Chemo is chemicals. So you're using chemicals, chemical molecules to alert our body, to alert it and kind of send an alarm mechanism, kind of sound an alarm that there is an intruder, there is an invasion, there is an injury, there is an infection. So whichever tissue is injured, whichever tissue is infected, the cells in these tissues will release some chemical mediators. Now, these chemical mediators can be things like interferons or even antimicrobial proteins or complement proteins, or they can be things like histamine and things like that. So a lot of different chemical substances can be released. Okay, what these chemical substances do, they attract the macrophages, the tissue macrophages or the wandering macrophages to the site of infection. So that is chemotaxis. Chemotaxis is basically using the chemical molecules to bring macrophages to the site of infection or injury. Not only macrophages, also the neutrophils. So any phagocytic cell basically. Once that phagocytic cell has come in the vicinity of that pathogenic organism or the microbe or the dead cell or whatever has triggered chemotaxis, then it goes into the next step, which is it must adhere, it must bind, right? So on the surface of our tissue cells and on the surface of our neutrophils and macrophages, we have lots of receptors, proteins, which bind with specific molecules called ligands. Okay. Usually for adherence, they will either use the part of the antigen that is being presented or a protein that is sticking out from the surface of a bacteria or a virus or whatever has actually triggered chemotaxis. Right? So that is the next step. Next step is adherence. Adherence means attaching with the pathogenic organism or with the causative organism that has initiated this phagocytosis response. Okay. Now it can be even your own body cell that needs to be ingested. Like sometimes, pathogens, they hide in our body tissue cells, like the macrophages of our body cells or things like that. So then these cells will become the antigen presenting cells. So they, in phagocytosis, sometimes the phagocytic cell has to bind with the antigen presenting cell itself. Sometimes it binds with the microorganism. Okay. So that binding is called adherence, attaching with the causative organism or the thing that has triggered phagocytosis, that is called the adherence. Binding of the proteins with, so ligands, let's call them ligands, because ligands and receptors, that's how it works, right? So when the ligands on the antigen bind with the receptors on our phagocytic cells, they initiate a signaling cascade. And if you remember how the signaling cascade works, the signaling cascade ultimately will be activating certain target proteins in our cells. Okay, so through the cascadic reactions, by activating the second messenger and then by activating the kinases and then activating the other target proteins, it will activate the cytoskeletal proteins in the phagocytic cell. So in the neutrophil or the macrophage, which has attached to that pathogen. Okay? Once that happens, the cell membrane of that phagocytic cell will start to fold around that attached particle. So wherever that attachment has happened, the ligand and the receptor binding has happened, around that, the cell membrane will start to fold. And that is what you see in here, right? So I'm just going to go through the picture in this slide. Excuse me. Sorry for that. So this is the pathogen or a microbe that has triggered chemotaxis. So you can see all these tiny chemicals that have been released by the microorganism, and that has activated this phagocyte to come to the site of infection. And now we can see the binding of the microorganism with the phagocyte, and you can see these folding of the cell membrane around it, right? So this is called pseudopod. Pod as in feet, right? So digits or fingers or feet. So pseudopod is false feet or false fingers or false limbs, you can say. So they kind of start extending around the microorganism and they form a vesicle around this organism. Now this vesicle containing the microorganism, that is called a phagosome, because it is a vesicle, so zome for the vesicle, and it is containing a phagocyte. So the pathogen, so phagosome. Okay, so phagosome, once it is formed, it is then brought into the cell. That is your third step, ingestion. So you have now brought the phagosome with the pathogen into the cell. Okay, next, this phagosome is carried to a suitable lysosome, which has digestive enzymes that will help to either kill the pathogen or inactivate the pathogen or neutralize it. Different ways in work on pathogen. Okay, so that is the fourth step, digestion. So digestion is basically when you fuse the phagosome with the lysosome of the phagocytic cell. So here you can see this is the lysosome of a phagocytic cell. So here the lysosome is mixing with the phagosome, and sorry, my mistake. The green one is the lysosome of the phagocytic cell, And this, of course, is the phagosome. So green lysosome is mixing with the phagosome. This is a step of digestion. So during this digestion, the lysosome will release its digestive enzymes, which will help to break down the cell wall or the cell membrane of the pathogen and break it into tiny particles, small digestible particles. Okay, many times these particles are recycled and reused if possible, and then the fragments or the things which cannot be reused will be removed from the cell through the last phase. So we'll talk about that. Okay, so during the digestion, the pathogen is broken down into smaller fragments or made inactive. Okay, the digestion continues till you can no longer reuse the particles that have been broken down, and that is the final stage of killing. So in the killing, the residual part or the indigestible material of that phagosome is then removed from the phagocytic cell to be removed from the body. Okay, so those are the 5 steps of phagocytosis. Doesn't matter where phagocytosis is happening, it is always going to follow these 5 steps: chemotaxis, adherence, ingestion, digestion, and killing. Right? So during digestion, there are a lot of things that can be produced. Some of these will be used by our cells to be recycled and used by our cells for their metabolic activities. Some of the things will be harmful to your cells, and so they will have to be removed from the body. So in this slide over here, it's actually showing you the things that are kind of helpful or harmful to your tissues. Okay. So once the killing happens, whatever is remaining in that undigested vesicle, that will be removed from the cell through exocytosis, and then it will be removed from your body either in the form of discharges or urine or stool or whatever, right? So but that is inactive form, so it's no longer infectious. It doesn't have the capacity to cause any further infection. Okay. So there are a lot of ways in which the microorganisms can actually evade phagocytosis. They can kind of stop phagocytosis or evade it. So there are different ways in which that can happen. Particularly, their own defense mechanisms, right? So some of the pathogens, they may have a protective capsule around them, which is kind of a protein coat or a thick protein layer that makes it difficult for them to adhere, to attach with a phagocytic cell. Okay. Now, in order to attach, in order for adherence to occur, you need to have those ligands on the surface of the phagocytics or the pathogen, right? So some of the pathogens may have capsules. So when they have a capsule, those ligands are not easily accessible. So obviously it's very difficult for them to bind with it. Sometimes the capsules are made up of a very sugary protein coat. So it's a very slippery coat. So they slide out when it's trying to attach, so that is one way. Another thing that the microbes can do is that some microbes, they produce toxins. They produce cytolytic proteins, like say for example streptococci, right? Now streptococcus is a bacteria that causes sore throat, very common, right? Most of the cases of sore throat, if you look at the bacterial sore throat or the upper respiratory tract infection, usually if it's not viral, then it will be a streptococcal infection, right? So very common. So streptococci produces a protein called streptolysin. Right? So it is a toxic substance, lysine, so breaking down, right? So it is something that is going to break down your tissue cells or your body cells or your phagocytic cells. So that's where it can evade phagocytosis. Okay. Sometimes they interfere with the lysozymes. They block the secretions of the lysosomal enzymes by producing these proteins or inactivating the lysosomal activities. Those are different ways in which, few of the different ways in which the pathogens can pain, psychosis. Usually your body will try to overcome these mechanisms, and it will have more than one mechanisms in place to tackle with these pathogenic organisms. Okay. Any questions? Okay, right. So then we can move on to the next one, which is the inflammation. Now, when we talk of inflammation, inflammation itself is a non-specific immune response, right? So it is something that your body is born with. It already knows how to do it. Inflammation is a response to an injury or an infection of any type in your body. Even allergic reactions will cause inflammation. Specific immune responses will also cause inflammation, right? So when we talk about inflammation, any inflammation is characterized by 5 major signs. They are called the cardinal signs of inflammation, or the basic signs of any inflammation. There has to be redness, there has to be swelling, there has to be pain, there has to be redness, swelling, heat. There has to be heat, there has to be pain, and there has to be loss of function. So these are the 5 cardinal signs of any inflammation: redness, heat, swelling, pain, and loss of function. So let's have a look at the mechanism as such to understand how these signs develop and why they develop. Okay. So here in this slide, the one that you're seeing, you can see that there are a lot of different causes for inflammation. To summarize, you can say that any type of injury to the tissue, any type of infections in the tissue, any type of allergic reactions in the tissue, any type of autoimmune or hypersensitivity reactions in the tissue will always produce inflammation. So you will see inflammation running pretty much throughout the immune responses. Most of the immune responses, first line, second line, third line, whatever you consider, basically there will be inflammation happening at some point in these responses. Okay, it is considered as a second line of defense because it is a non-specific type of a defense. Okay, but it is seen in specific responses also. Okay, so 5 signs I've already told you: redness, heat, swelling, pain, and loss of function. So let's have a look at why this happens. Okay, I would have loved to use some whiteboard, but I can't at the moment. Okay, I'll figure out if there's a way to use it. Okay, so talking about the inflammation. So first step in any inflammation is release of chemical mediators. So whenever there is a tissue injury or infection in the body, the injured tissue or the infected tissue of your body, the cells of this tissue will release some chemical mediators, some molecules, which will alert our body about the condition, about the injury or the infection. These chemical mediators, generally, they are released by the mast cells in the tissues or by the injured cells of the tissues. There are a lot of different kinds of chemical mediators. At the point, you don't have to worry about remembering their names. But just to put it into perspective, some of the chemical mediators that are released are things like histamine, or things like, say, kinins, bradykinin, and things like nitric oxide from the blood vessels, etc. Right? So the injured tissue releases chemical mediators which will initiate vasodilation. Vaso is blood vessel. So dilation of the blood vessels. So things like histamine, they work on the smooth muscles of your capillaries and they stretch these smooth muscles, they relax these smooth muscles. So there is dilation of the capillaries. And as the capillaries dilate, the endothelial lining, the cells in the capillaries which are close together, they separate. Now when they separate, there is more gap, right? So that allows the fluid, the proteins, the cells to migrate out of the blood vessels. So that is the reason why you will have swelling. Swelling is collection of fluid in the tissue. We'll have redness and heat from vasodilation, because when the blood vessels are dilated, the quantity of blood flowing through them increases, so that causes the redness as well as the heat. Okay, so vasodilation and increased capillary permeability. Why does the capillary permeability increase? Because the capillary cells are being stretched apart due to the dilation, right? So vasodilation and increased capillary permeability allows the fluid and the plasma proteins to leak into the tissue spaces. Along with that, they also allow the cells, phagocytic cells. Now, when we talk about the phagocytic cells, we are primarily talking about the neutrophils over here, right? Neutrophils and macrophages. So it will allow these cells to escape from the capillaries into the tissue spaces, right? That is called the chemotaxis. So you can see the migration of phagocytes happening. You can see here, this is the blood vessel, right, in this diagram. So this is the phagocytic cell, the neutrophil or the macrophages, and this is how it is immigrating. It is squeezing through the gap that has been created because of the stretching of the tissues. Okay. Now this is again. Please go. Okay. So here this is a phagocytic cell. This is also a phagocytic cell, probably a macrophage or a neutrophil, and they are releasing the chemical mediators to target the microorganisms. So during this second phase of inflammation, there will be same things that we talked about in phagocytosis. Same thing will happen. Any tissue will be clear of any positive pathogenic organisms or microorganisms that have entered or invaded the tissue spaces, right? The final step is the tissue repair. So once the phagocytes have been cleared from once the pathogenic cells have been cleared, pathogens have been cleared from the tissue, like here, the tissue repair will happen. Now, when we talk about tissue repair here, this we are talking about the deep wound healing that you would have learned in biology 1 when you were doing the integumentary system, right? So in the deep wound healing, the first thing that should happen is, of course, the inflammation, and then the second thing is the basal membrane, the basement membrane of the epidermis or of the epithelial tissue needs to repair itself, right? So during the tissue repair, the cells in the basement membrane, the cells on top of the basement membrane, you can say in the basal layer, they will start to enlarge and move towards each other. They will continue to move from both the directions till the cells from both the sides touch each other. That is called contact inhibition. So when they touch, they stop migrating, right? Once the contact inhibition occurs, then these cells will start to divide. They will undergo mitosis. And as they go through the mitosis, they will fill up the gaps that were created because of this migration of the cells. And once the basal layer is intact, the rest of the layers will continue to develop, like just like in the skin, right? So they continue to multiply and they continue to create the other layers of the. Sorry for that, my kids are in the room. Okay, so they continue to repair the tissue. Okay. So that is basically the inflammation. So during this inflammation process, in the initial stages, you will obviously see the heat and the redness and swelling happening. Now, because the tissue is stretching, while histamine is causing all this from like the vasodilation and increased capillary permeability, there is also something else that is happening. In the mast cells of your tissues, in the injured cells, not the injured cells, the mast cells of your tissues, there are specific molecules, there are specific chemicals which are called like the prostaglandins and the leukotrienes, right? So these are the things. So prostaglandins and leukotrienes, they are responsible for continuing the effects of inflammation that is happening, and they are also responsible for pain. Okay, a lot of pain comes from the stretching of the tissues around the injured tissue. So those stretching of the tissue activates the pain receptors, nociceptors in the tissue, and triggers that sensation of pain. And a lot of it also comes from the molecules like prostaglandins and leukotrienes. Okay. Because of the pain, obviously, you will have restricted movements and you will not like to move the tissue or the body part, and that causes a loss of function. Okay. So pain can result from a lot of things. Pressure that is created because of the stretching of the tissue due to edema, or release of chemical mediators like prostaglandins and leukotrienes in the tissue spaces. Now these prostaglandins and leukotrienes, they are produced by the mast cells themselves. So we don't go into details of these at the moment, but you will study about inflammation in a lot more detail when you go into pathology. So you will study the pathway, how mast cells produce prostaglandins and leukotrienes from arachidonic acid in their structure. Okay, so don't worry about them at the moment. Any questions? No. Okay. Right. So then we come to the fever. Okay. So fever basically is increase in the body temperature. So it's basically increase in the thermostat of the body, right? So if you remember the functions of hypothalamus, one of the functions of your hypothalamus is to regulate your body temperature. So it is basically to increase the body temperature when required and control the body temperature, right, according to the environmental temperature. Now, one of the factors that increases fever is the release of. Please go. So one of the factors that just bear with me for a minute. I will have to do something about the kids. Okay, please go out. Okay, so the pathogens that have entered your body or the injured tissue will release chemicals. Again, these are chemical mediators, right? They are called pyrogens. So release of these pyrogens will help to reset the body temperature by resetting your hypothalamic thermostat. Okay? So in a way, we can say that fever is a good sign. You know, it's good to have fever, because when you have fever, it means that your body is actively working through the second line of defense. Increasing the body temperature helps to mobilize the pathogens. It helps to localize the infection. It helps to fight the pathogens by activating it to release a lot of other chemical mediators as well. You know, so it's a basically a good way, good response of your body, because it helps to increase the effect of interferons, which are the antimicrobial substances. It helps to speed up phagocytosis, causes vasodilation, so it helps with inflammation as well. You know, so a lot of beneficial aspects of fever. Okay, so fever, antimicrobial substances, inflammation, and phagocytosis, all of these, and of course natural killer cells. So they are all second line of defense. They are all things that are basically innate. You are born with it. Okay, any questions? Right. Then we come to the last part of this session, which is basically an introduction into the specific immunity. I will be talking about specific immunity in much more details in the next webinar on Wednesday, right? Because that's when we are going to discuss a lot more about the specific immune response. But I just want to talk just a little bit briefly about what is the difference between a specific immune response and an non-specific immune response or an innate immune response. When we talk about specific immunity or adaptive immunity, there are two main properties of this type of response. Okay, firstly, these are very selective, so they're very specific to a particular pathogen. The way your T cells and B cells might respond to a chickenpox virus will not be the same as it would respond to a tuberculous bacteria or even to any other virus, you know. So they are very specific for that particular virus or pathogen or a bacteria. Okay? And secondly, whenever there is any specific immune response, there will always be a memory of that response. So your T cells and B cells will keep a record of the events that have happened and the type of antibodies or type of responses that they have used for the future reference, so that we can use that as our future reference, and we can have a stronger response if we were to be exposed to that particular pathogen again in future. Okay? So we'll talk about adaptive immunity in a lot more details in the next sessions, coming sessions. Okay. So there are different types of adaptive immunities. You have the cell-mediated immune responses, which is mainly carried out by the T cells, the T lymphocytes, and you have the antibody-mediated responses, which are carried out by the B cells. Okay. So let's stop here. In the next session, I will continue with the maturation of the T cells, B cells, and how those specific immune responses take place, right? So that's going to be a recap of all of this in the next session, which is on the Wednesday. Now, anyone has any questions from the tutorials? Have you all done the tutorials yet?
Not yet.
Okay, I did have a question from the tutorial from the last session, so I'm just going to share with you. One of the questions from the session 7 tutorial was about the follicular cells, right? So somebody had asked me over the LMS about what are follicular cells and where they are found. So just let me go back to the code and code tutorial. Okay. Just going to open that up. Okay, so this diagram is in your textbook already, so you can use a textbook to label the diagram. Okay, but when we talk about the follicular cells, now here in this table, it's asking you about the location of the follicular cells in the lymph nodes. Okay, so whenever we talk about a follicle is a group of cells, is a cluster of cells. Okay, follicular in case of lymph nodes, follicular cells are located in the outer cortex of the lymph node. They're a part of your germinal center. So within the germinal center, you have the collection of the B cells and you have the dendritic cells. So these dendritic cells within the germinal center, within the collection of the cells, that they are called the follicular dendritic cells. Okay, and what do? Their main function is to present an antigen to the B cells, so that they can actually so they present the follicles to the B cells, so they can actually create antibodies against that particular antigen. Okay, to find. So in a way, they are helping the B cells to convert into a plasma cell. Right. So that was from the previous tutorial. So when you go through the tutorial for this session, I'm just going to go back to the current session, which is session 8. And this is a tutorial associated with session 8. Okay. Have a go to in completing these tables, and if there are any questions, do let me know. Now, these are part of your textbook, so you can find them in the textbook for labeling purposes. So you can use that, right? I have tried to cover all the aspects that are included in your tutorials already. But if you still have any questions, feel free to write them to me. Yeah. Right. Okay. So that brings us to the end of this particular webinar. Any questions, anyone?
Me? How do you do that one?
Right.
Is that the blood?
Just going to stop the recording.